In a lot of ways, you can think of cities as one of the largest unplanned experiments of all time.
There can be really small differences that have very large biological significance.
Cities are places, we call them extreme habitats really. They are places where there is a lot of opportunity and at the same time there's also challenges.
As our cities spread, how will nature respond? Will plants and animals dwindle, or will they adapt to urban life? And what kind of new encounters will we see in the city? In the historic French town of Albi, biologist Frédéric Santoul keeps an eye on his catfish. In 1983, fishermen released these Eastern European fish into the River Tarn. Today, they're at the top of the river's food chain.
This is a fascinating species because we know so little about them. There are many myths that people believe, even that they eat dogs. There are many stories.
The biologist is interested in the behavior of the large fish that circle the reservoir's basins.
We work with fishermen to tag the fish. They contacted us after observing very strange behavior in the fish here in Albi.
The man-made landscape of the city fosters new encounters of species.
The pigeons have never had to face predators from the water. Instead, they scan the sky for birds of prey.
The pigeons approach the water to bathe and drink. Sometimes a bird misses the narrow strip of safe ground and touches down in open water.
The catfish don't really see the pigeons, but once they sense the birds' movements in the water with their barbels, then they strike.
This new hunting behavior of the catfish was observed by the biologists for the first time in 2010. For some catfish here, pigeons now account for up to 40% of their prey.
There's suddenly this ecological interaction which allows for evolution to start to improve the bird catching ability of the catfish and also to improve the escape ability of the pigeon. So you can expect that all these new interactions are also causing new evolutionary dynamics.
Dutch evolutionary biologist Menno Schildhausen researches the adaptation of wildlife to the city. Darwin's theory, he believes, has gone urban.
Urban evolution is evolutionary change, so really genetic change in wild animals and plants in cities. It's all about understanding how species will be able to survive in this very human-dominated context.
Cities are Homo sapiens' most extreme intervention in nature. With concrete and steel, we create new landscapes and alter the face of the earth. Already, most people live in cities rather than in the countryside. How does this influence evolution, the development of new species? What selection pressures does the city create? A summer evening in the Dutch capital, Amsterdam. In the Vondel Park in the centre of the city, biologist Menno Schildhausen uses a light trap to catch insects. He's leading a citizen science project to explore urban nature.
For insects and for some smaller plants, the diversity today in cities seems to be higher than in intensively managed agricultural areas. Today, agricultural land is so intensively managed, and the last bit of production is squeezed out of every square meter of surface area, that there's no space for nature anymore in the countryside. And at the same time, cities get greener. People pay more attention to nature and to urban nature. So it's actually becoming a very rich environment with a higher biodiversity than outside of the city.
But overall, we're rapidly losing biodiversity, both within and outside our cities. For insects, the declines are particularly severe. In the Swiss Alps near Zurich, scientist Florian Altermatt has set up his light traps. Ever since humans began to light up the night, billions of nocturnal insects have been dying off every year.
For such a species, it's problematic if it's attracted by light, because then it cannot use the few short days it has as a moth to lay eggs.
Light pollution is one of the major threats to moths. Scientists are now even speaking of an insect apocalypse.
I think the declines we're now seeing are already quite worrying. Studies show a 60 to 80% decline in biomass, sometimes even in nature reserves. These are incredibly large numbers. In my childhood, I used to observe moths like these. I would set up this trap next to my parents' house and attract moths, actually in quite large numbers. I don't think I'd find many today.
But might insects be capable of adapting to life in the perpetual light of our cities? That's what Florian Altermatt wanted to find out. His test subject? The spindle ermine moth, whose caterpillars develop on the European spindle tree.
Actually, it was a coincidence. While I was working on my PhD thesis, every day I walked through a park which had those European spindle bushes. And I noticed that there were these caterpillars, these moths, which must have lived there for years in a city park with permanent light pollution. And then I thought I could just collect them, raise them, and test how much the adult moths are attracted by light.
With his experiments in 2006, Altermatt pioneered research into urban evolution. He released the moths in a darkened room. The next morning, he counted how many had flown into the light trap.
The results showed a difference. About 20% fewer urban moths flew into the trap. I was very surprised. It was widely known that moths are attracted by light, some more than others. But these differences have always been observed between different species. Seeing variations within a single species, that we'd never seen before.
The experiment clearly demonstrated a hereditary adaptation to life in the city. Direct proof of urban evolution. For Dutch biologist Menno Schildhausen, the findings confirm a larger picture. In Amsterdam, he and his group of citizen scientists debate whether we might soon observe even more and greater adaptations of animals and plants to this city.
We see that evolutionary processes are starting which will eventually or could eventually produce new species that are specialized on living in the city.
For Menno Schildhausen, it's not if, but when.
Every organism that lives in the city will show this urban evolution, these rapid changes in their behavior, in their physiology, in their appearances, to optimize their life in an urban environment.
But what elements of urban landscapes prompt wildlife to adapt? Evolutionary biologist Jason Munchie South is an expert on animals found in the parks of New York City. For years, he's been studying how rodents adapt to the city. Along with human immigrants from Europe, rats also voyaged to the New World. Today, they roam the city in subway tunnels. Most native rodent species, however, don't dare try their luck crossing town. This distinction sparked the scientists' interest.
I used to be a tropical biologist, but then I moved to New York City for my first academic job after graduate school, and I decided I wanted to do some local work that would be interesting to the people of New York City and to myself. And I found out that there were these small mammals living in essentially islands of forest in the city, and I thought, that's interesting. Nobody's really ever looked at these. Are they becoming genetically different from mice outside the city? Are they adapting? And that's how it all started.
Central Park opened in 1873. It still hosts animal species that lived here long before the city was built.
Right now, we're in the middle of Central Park. We're going to be traveling to the north end of the park where there's a very nice forest called the North Woods. And there we'll be setting out traps, hopefully to capture white-footed mice. One of the things that inspired me when I first started this work is if you look at a New York City subway map, you see the subway lines, but then there are these large green shapes, rectangles and ovals and so forth, that are the parklands. And they've put those on the map so you know where they are, but you also see that they are almost like a chain of islands that are scattered in this sea of concrete and roads and buildings and 8.5 million people. So in a sense, if it's a species like a mouse that can't leave the forest, cross neighborhoods and buildings and roads and make it to the other patch, it is essentially the same biologically as if they were on an island in terms of them not being able to move and spread their genes with the other patches. And these urban patches, once they become sufficiently isolated, operate like a mini Galapagos, and maybe driving the evolution of many species that are, stuck there now.
The evolutionary biologist is investigating whether the white-footed mice actually develop in distinct ways in each of the various parks.
So this would be a really nice spot for white-footed mice. They like to move next to logs, so they're not completely out in the open. They might actually even be living inside this log where it's rotting or in holes underneath the log. So this is pretty much the ideal spot.
This forest is encircled by the Big Apple. Have the mice already adapted to this unique environment? What traits do they need to survive here?
No shortage of good trapping spots. Later I'll be going to one of our more suburban, almost rural sites with a larger, more intact forest, less urbanization, and I'll be setting out, an equal number of traps with the hope that we catch mice there as well.
Jason Munchie South will search within the animal's genetic codes for the markers of life in the big city.
I think what's been most interesting to me is thinking about how the things that we are all doing in our daily lives, where we put our garbage, what we're choosing to eat and what we generate as waste, where we choose to live, how we choose to go to work or out to a restaurant or something, all of these things we are doing are now influencing other species in a way that we're just starting to understand.
Though it's not only animals that adapt to human intervention in the natural world, so do plants. In southern France, the yellow-flowered Crepus sancta is being studied by biologist Olivier Chaptou.
Crepus sancta is a very common species in the Mediterranean region, a kind of Mediterranean dandelion from the same family. And its essential advantage as a model is that it produces two types of seeds, large ones and small ones.
The small wildflower produces both lighter seeds with parachutes allowing them to glide and heavier seeds that simply fall to the ground.
I'm interested in the process of adaptation to an urban environment, and in particular, what happens when a species first arrives in a city. It has recently colonized certain areas of Montpellier, and in my comparison between rural and urban populations, I focus on the traits related to seeds.
The idea of studying the adaptation of the modest plant to the city came to Chepteau almost by chance. When he came back from abroad, he noticed the inconspicuous flowers growing in the city.
I had left Montreal in the middle of a blizzard. I got the plane to Paris, and when I took the bus to downtown Montpellier, it was sunny with a pristine blue sky. And then I noticed there were Crepis sancta flowers everywhere in those tiny urban patches. It was then the intuition hit me. I knew there was something to discover here, something to uncover.
Was the plant already adapting to the city? Which aspects of the city would drive that change?
The predominant mineral in cities, especially in European cities, is concrete. Concrete exerts a powerful fragmenting force on plants, driving them to ever-shrinking refuges in these habitats.
Sometimes the city's constraints on a plant's habitat can be extreme. How will evolution respond?
I'm looking at how urban fragmentation will modify the dispersal traits of this species. I expect plants that produce more of the larger seeds will be more successful at reproduction in urban areas than they would be in the countryside.
The heavier seeds face less risk of being swept onto the asphalt. And indeed, the biologists discovered that far more plants in the city produce the heavier seeds and are thus better able to survive, a difference of 15%. But what most stands out is the speed of this adaptation.
The evolution we have seen has taken about 15 years. This is extremely brief. It was the first demonstration of such a rapid evolution of seed traits for plants. And that's due to this highly fragmented composition of the urban environment.
Genetic changes occurring at such a rate have long been considered unlikely, even impossible by science.
I think Darwin would have been amazed by the fastness by which these changes take place. He was sort of underestimating the power of natural selection himself. He said that you could never see any of these changes in progress. You cannot actually observe them. You can only deduce them from the fossils, from the patterns that you see in nature. He said that evolution is too slow to see it happening in real time. And the fact that now today, especially in cities, we see these changes taking place under our eyes in the streets where we live, right around us, I think Darwin would have been thrilled by that.
In Los Angeles, the UCLA campus also nurtures species that have migrated from surrounding areas to the city. Evolutionary biologist Pamela Yeh studies the dark-eyed junco. The mountain bird has only been settled in California's cities for a few decades. Pamela Yeh began her research in San Diego more than 20 years ago. Today she's investigating how the once shy bird manages to survive in bustling Los Angeles.
So this is a junco nest. Typically in the mountains they make all of their nests on the ground. But up here, we have a good percentage of our nests that are up in the middle of, branches of trees or up on the buildings, higher up off the ground.
Darwin himself was fascinated by the evolution of birds, but until now, biologists couldn't observe their adaptation to new habitats in real time.
They have this very famous study on Darwin's finches in the Galapagos, and they can show really large biological differences in terms of mortality, like who's surviving and who's dying, by a millimeter or less of a beak depth or a beak length or a beak width, right? And so I think there can be really small differences that have very large biological significance.
To track such subtle changes, Pamela Yeh and her team, led by PhD candidate Ellie Diamond, first have to catch the birds.
So the nest is just over here. You can see it in the grate. There are 5 chicks up there, 9 days old. and the parents are going back and feeding them.
Might recorded birdsong jolt the bird into rash action?
So we're going to play a playback, a territorial junco song, and it's going to go to that speaker there, and hopefully the male who seems like he's feeding his chicks right now is going to fly over, try to check it out, maybe go for the speaker and hopefully get caught in this net or in this net. We'll see. Okay.
The call of a rival bird. The male, intent on driving it off, rushes over.
Oh, there is a junco in the net. Guys, that was easy.
The team tags and measures the bird and collects DNA samples. Back in San Diego in 2003, Professor Ye had discovered a change in the appearance of the city junco's tail feathers. Now her team wants to see if something similar is going on here.
Spreading his tail so you can see the white on his tail there.
One of the main differences that we found is that urban juncos had much less white in the tail feather than the populations in the local mountains, which had a lot of white. And what we wanted to know was then, well, why is it there's this huge difference that we're finding between the mountains and the city juncos? What's the reason for that?
Evidence suggests the white tail feathers serve an important signaling function in competition and partner selection by the birds.
Cool. Success. All right.
What new demands does life between motorways and buildings make on birds to change their mating and territorial preferences? The case of a species adapting its colour to man-made environments was first observed by natural scientists during industrialization, even before Darwin put forward his theory on the origin of species.
The peppered moth is a species of moth that comes in two forms, a light-colored form with light wings and one with dark wings. And the dark-winged form was only first found in England in the Industrial Revolution in the middle of the 19th century. It began to appear and suddenly it started to become more and more common over a relatively short period of time in areas where there was a lot of air pollution.
The smoke from the chimneys coated the white birch bark with dark soot. The white moths thus became easy prey for birds. The new darker moths enjoyed superior camouflage.
People didn't really understand what was going on. They thought that the female moths maybe saw that the environment was getting darker and somehow that became imprinted on their offspring. I mean, that's the way people thought about genetics and heredity in those days.
The conclusive evidence connecting the spread of the dark moths to coal soot was only found in the 1980s. The air was getting cleaner again and the white moths returned.
Evolution can proceed fast if the environment changes very dramatically, which means that a lot of individuals will die and a few will survive, the ones that have some genetic characteristic that makes them survive better, and they will then produce the next generation. And they may also have some adaptations which then spread so that evolution can proceed fast.
The colour change was a simple but effective adaptation. It only required a mutation of a single gene. But what if man-made pollutants substantially distort the biochemistry of organisms? In the 1970s, the water at New Bedford Harbor near Boston was severely polluted with PCBs. The US Environmental Protection Agency, EPA, wanted to know just how bad the pollution really was.
The original focus was on what must be wrong with all the fish that live in that harbor because of the toxic chemicals. Instead, we came here trying to understand what must be right about those fish that could survive here. So they've become a natural experiment for us to study how animals can adapt to toxic human-made pollutants.
Terrific. Just what we're looking for. Let's get them into a net, bring them back to the lab.
Diane Nachi heads the EPA lab in Narragansett, Rhode Island. In their breeding facility, the scientists want to unravel the mechanism that allows this population of a killifish species to survive in the PCB polluted water of New Bedford Harbour.
So let's see if they left any eggs for us.
They plan to compare eggs from the New Bedford Harbour fish with those of a fish population from a cleaner site.
Let's start a test and see what they do when we expose them to chemicals.
This killifish species occurs all along the North American Atlantic coast.
The killifish has been a favorite of biology literally for centuries. They are quite common. They are non-migratory, so they reflect their local environment. And each population is unique in that it is genetically different. It is adapted to its local environment. So it gives us opportunity for lots of studies.
The researchers need to observe the development of the fish embryos in the egg in order to understand at which stages the environmental toxin disrupts the animal's biochemistry, or not.
So we'll look at the rate at which the embryo is developed and certain features that we know that PCBs can disturb, like a proper development of the heart, evidence of proper development of the circulatory system, and proper body size.
Why are these particular fish able to resist deadly environmental toxins?
It's important to understand which species can and which cannot adapt, because we want to forecast, we want to predict what the environment will look like in the future, whether the ecosystem services that we rely on today, like pollination, clean water, etc., can still be relied on. And for that, we're going to need adaptation of all kinds of animals and plants to this new world that we are creating.
In California, some city juncos have less white in their tail feathers than their mountain peers. Which selection pressures are driving this evolution? What is the function of this pigmentation?
And one of the main purposes appears to be in dominance interactions between individuals, where typically the more white that you have, the more dominant and the more aggressive you are. And you're successful at winning competitions. If you're fighting with another one, often the one that has more white wins.
So we're going to do some aggression playbacks around Dixon Court.
All right. Good luck today.
Yeah. Thank you so much, Pam.
Okay.
We'll
See
keep
you
you up
soon.
to date.
Okay. Bye-bye.
To learn more about the adaptation of the birds to the city, PhD student Ellie Diamond aims to scientifically measure their aggression levels here in Los Angeles.
So we're essentially setting up an arena to watch the bird. Well, they're in the car. It's fine. This is Boris the fake junco. And it's the visual stimulus for the bird. He's just going to sit here, and there's going to be a song playing from there. And the bird is going to think that this is a real junco, and it's going to hear the song that it's singing, and then hopefully come and think that there is an intruding bird on the territory, and we'll see how it reacts to that bird.
Juncos are territorial birds. From a male's response to a stranger in his territory, the researchers can gauge his aggressiveness.
Ready?
Yeah. Sorry, starting now.
Okay. I think I hear him. Oh, he's there. Yeah, that's him. He's in the tree.
This angle you can.
Yeah, let me try. He's still singing.
Turn around.
Where is he? Oh.
He's down. Up.
Oh, he's in
He's
three.
entered.
Shortly before the end of the 15-minute trial period, the male stalks the intruder closely.
He's hiding behind some grass. He's not gonna, is he gonna knock it over?
The researchers stopped the playback to end the experiment. The male displayed some aggression, but not a great deal. Why do the city birds seem to lose their drive for competition?
What we think is happening is that their selection for less aggressive birds who are better mates, better parents, because you can likely increase your fitness so much more by taking care of the chicks that you do have rather than looking for extra pair copulations and extra pair fertilizations.
With the longer summers in the city and abundant food from watered greens, the birds can maintain more nests per year than in the mountains. Competition for additional mates and territories no longer seems worthwhile.
There's a lot of reasons why these birds shouldn't be successful in urban areas, and yet they are. So I think understanding why they are and understanding the mechanism, the processes underlying that, can tell us something about how do we encourage other species to also be successful.
What are the factors that allow individual species to adapt to the city? Are parallel developments taking place in cities worldwide? At the University of Toronto, Mississauga, evolutionary biologist Mark Johnson pursues these questions.
In a lot of ways, you can think of cities as one of the largest unplanned experiments of all time. The problem is there's very few organisms where you could study adaptation to urban environments on a global scale, and white clover is one of those very few organisms where you can actually do that. And so now, this then becomes the model to understand whether organisms in general can adapt to the convergent environmental change associated with cities throughout the world.
Researchers across the globe are working together in this study. Evolutionary biologist Stefan Greiner and his team are collecting white clover in Berlin. In cities, the plant faces a different habitat. Temperatures are higher than in the suburbs and the countryside.
What you can expect is that as humans create new environmental conditions, life will adapt. And to be able to show that on a global scale, that's a real scientific benefit. It has to be done. That's why we're dedicating our free time to this project.
As they proceed from the countryside to the city centre of Berlin, Greiner and his team collect specimens at 30 locations. That means the data will be broad enough to compare with that from other global cities. In the shadow of the television tower, they find their final samples.
That's it. All done.
In all, we have 168 cities right now, and over 250 collaborators all working on the same project together. There's never been a collaborative project on evolutionary biology of this scale. And so this is the largest collaborative project in evolutionary biology ever.
So, is clover developing in the same way all over the world into a kind of global city clover? From the vast set of data, the researchers hope to find an answer. In the grounds of a research institute north of New York, geneticist Jason Munchie South wants to catch white-footed mice to compare their DNA with that of those in the city. But it's not easy.
Go to the lab. It's good. Okay. So this is a trap that was opened. It didn't catch anything, obviously. Oh, that's a toad. I thought it was a mouse. Nice day for other wildlife, I guess. We were really surprised that almost every park was different from every other park. It's almost to the point where you could take a mouse from one park, give it to our lab, and we could just look at a small segment of its genome and tell you where it came from. That's how much they had changed just randomly over time from being isolated. And that's when we started our current studies, looking at over 20,000 genes to see what genes and potentially what functions change when they adapt to living inside of New York City. Oh, there's one. The first white-footed mouse of the day.
For the new study, Jason Munchie South and his team have already caught more than 100 mice and analysed their genetic compositions. They try to take their samples as gently as possible so they don't hurt the animals.
When we take a genetic sample, in this case, we will be using this small tool. It's like a paper punch, but for tissue, and we store that for genetic analysis. And we want to be able to tie that tissue sample to a location because that's important for understanding how they vary when they're in a more urban or a less urban population. So now they're pretty immobile. This is a male. It's a young male. All right, why don't we take the ear punch and we'll start on the other one.
Hair isn't suitable for comprehensive genetic analysis. After collecting the samples and some measurements, the scientists release the mice. Genetic analysis can reveal the evolutionary trajectories of the mice. They point to a variety of physical and behavioral changes spreading among the animals, each of them unique to the challenges of each city park environment.
So we're starting to fill in our gradient really nicely. So here are the mice we have today from the Calder Center, and you can see it's right in between highly urbanized New York City and then all these sites we have up here and one out here. So Central Park seems to be our most distinct population to date. It makes sense, yeah. Most urban, probably the most isolated. So if you took a mouse from Central Park, some of its genes will be different from a mouse outside in the countryside in a big park somewhere. Right. So for this one particular
The food supply in Central Park, much of it human food waste, might have triggered a genetic response.
Other places in the city near Central Park. And then once you get... So what we've learned so far is that one set of genes that are changing in the city have to do with metabolism. So these white-footed mice are eating things, then they have to digest them and assimilate the nutrients. And we know it's evolution because a heritable change in DNA sequences is evolution.
Central Park mice seem to have genetically altered their metabolism to better digest fast food.
It raises several broader questions about what we are doing as a species. As we modify the Earth's habitat for our needs, how are we changing the future of other species? Not only are we affecting them, but we're changing what they will become in the future.
In Narragansett, Diane Nachi and her team are investigating how the different fish embryos exposed to the toxic PCBs have developed.
Come in. Hi, how's it going? Really well. So this is the study that's comparing Squirtin Creek and New Bedford Harbor exposed to PCB 126. So this one is the group that was treated with PCBs, also from the clean site, Squirtin Creek. And as you can see, the PCBs have had a pretty dramatic effect on the development, which is what we expect with these very toxic chemicals.
Yeah, and in my experience, when I see this constellation of anomalies, it's absolutely lethal. There's no way that an animal would even hatch, never mind survive after hatching, if the heart is not functional and the blood is essentially not circulating around the body. So let's take a look at that biochemical endpoint to see if they are also responsive at the biochemical level.
Using a special contrast agent, the scientists can trace enzyme activity in the unhatched fish.
So you can see that the substrate is fluorescing in the bladder, showing that this enzyme system is working, and we're getting the expected metabolites in the bladder.
That's a very dramatic demonstration of enzyme activity in a living organism.
Mm-hmm.
The active enzymes reveal how the organism tries to break down the toxin, but perishes in the process. Then the team observes how the offspring of the fish from New Bedford Harbor have developed.
Okay, so these are fish from New Bedford Harbor that were exposed to the same level of PCB that we were just looking at. As you can see with this embryo, it doesn't seem to have any effect. The heart is still beating normally and healthy, and it's developed really well.
That looks like an embryo that's about ready to hatch.
Some of them actually already hatched.
These fish should be dead, poisoned by one of the most lethal environmental toxins. But life, it seems, has found a way.
One thing we know about this class of chemicals is that in all vertebrates, including people, it turns on a certain enzyme pathway. So a normally responsive person, or in this case a fish, should have that enzyme system turned on if they were exposed to PCBs.
The contrast-enhanced image shows how the enzymes that normally respond to the toxin remain silent.
So in this case, I see very little that's glowing brightly. It is a dramatic visual difference that suggests that that enzyme system is broken in the New Bedford fish.
The killifish from New Bedford Harbour have changed their metabolism. The poison can no longer harm them. But which genetic modifications lead to the fish's toxin resistance? That's what geneticist Mark Hahn of Woods Hole Institute wants to find out. Could this be a key to understanding how nature might resist human interference? In the laboratory, he uses the CRISPR-Cas method.
It's an incredibly powerful way to modify the genetics of an experimental fish like this, to ask questions about the roles of certain genes, and in fact the roles of even single amino acids in the protein can be investigated with this CRISPR-Cas method.
To test their assumptions about the resistant killifish, Mark Hahn and his team experiment with zebrafish.
I want to find out exactly what are the changes in those genes, and to be able to actually zero in on the specific molecular changes that are responsible for the resistance, and to be able to recreate that in the laboratory to actually prove that that's the mechanism of resistance.
They're inserting portions of DNA taken from the resistant killifish into embryos of zebrafish.
Hey Mark.
Hey Neil.
How are you?
Good.
Good.
Which ones are these?
I'm injecting AIP exon 2.
If the zebrafish growing from these eggs exhibit the same resistance, they will have found the responsible genes.
Here we are interested to study a function of a gene known as AIP. So we are trying to delete this gene in this particular species and then try to study what's the function of this gene and whether that will alter the resistance to PCBs.
With these experiments, science is venturing deep into the source code of creation. The scientists believe this research could yield the secret of life's ability to adapt to the most extreme conditions. And this knowledge could also help other creatures to adapt and survive in a rapidly changing world.
I think we will understand the extent to which we can extrapolate our knowledge from the killifish system out beyond to other fish and even other vertebrates. So a broader understanding of the toxicology of pollutants and how that will impact the natural world, how we can understand what will be the most vulnerable species.
At the Max Planck Institute in Potsdam, the research team processes the clover samples from Berlin. Their goal? To find cyanide. Clover plants that produce cyanide are better protected against predators, but are less able to tolerate cold. It's warmer in city centres, so this clover might be more common there.
This is a qualitative test. We use it to indirectly detect a specific gene that generates this cyanide. So these are both rural?
Yeah, they're both still rural. Greiner and his team send their results and other clover specimens to Mark Johnson in his Toronto lab.
Hey gang, how's it going?
Good.
Beta, how did that extraction go yesterday? Yeah? So remind me, this is Berlin and Buenos Aires.
The team prepares the clover for gene sequencing. But the cyanide values taken by the team in Germany should show whether the clover has adapted to an urban existence already.
Okay, Beta,
Did we get the data from Berlin?
Yes, we did.
Okay.
On the screen here, it is. So
Great.
I think it's really good.
Okay, James, have you had a chance to look at the data from Berlin? So we can understand how the environment's changing from downtown Berlin through the suburbs and into the rural areas?
We got it.
All right, so let's take a look at. So Berlin's one of the cities where we see white clover adapting to urban-rural gradients.
Yeah.
Nice. And so now we're at about 33% of cities where white clover adapts?
Maybe 33.2 now. But yeah,
Fair
it's
enough.
about 33%.
Okay. So then next, I think what we're going to have to do is figure out what are the drivers, the environmental drivers of this adaptation. So that's really cool.
In Berlin, plants from downtown are more likely to produce cyanide, as is the case in a third of the cities surveyed so far. An indication of parallel evolution?
Some of the preliminary insights are fascinating. So it really looks like, regardless of where you are in the world, whether you're in Europe, North America, Japan, China, Australia, New Zealand, we see the ability for this humble white clover to adapt to these cities.
In the warmth of the city, cyanide-producing clover stands a better chance of survival. But to survive in the city, all organisms must adapt to higher temperatures, what scientists call heat islands.
In cities, humans and their machinery create a lot of heat, and we have a bubble of hot air in large cities. And a city of more than 1 million people can be 7-8 degrees Celsius hotter in the center of the city than outside of the city.
This, Menno Schulthausen believes, also influences the evolution of the white-lipped snail. Their shells come in many shades, from brown to pale yellow. A single gene determines the color.
So they basically carry their genes on their back. The shell color determines the internal temperature of the snail to some extent. The difference in temperature inside can be 2 degrees under the same conditions, and that could be just the difference between life or death. On a hot summer day, and you know it was 40 degrees in Amsterdam a few weeks ago, it could be that some of these yellow snails survived, but many of the brown ones died because they got too hot, they overheated, and they died.
But will statistics confirm this hypothesis?
Snails are evolving towards more yellow shell coats. So the plan is now to just add some data to the data set. So let's go.
In order to collect and evaluate as many snails as possible throughout Europe, Menno Schulthausen is helped by volunteers.
You know, you don't have to go to the Galapagos to study evolution or become a paleontologist. It's happening everywhere, all the time. It's a continuous, very normal biological process.
This time, the volunteers find only a few live snails, but empty snail shells can also provide data. The snail shells are photographed and added to the database with an app that anyone can install on their mobile phone.
And that's it. If we're looking at the adaptations of urban animals and plants to the urban heat island, which of course has been happening more rapidly than global climate change, we can probably predict what's going to happen globally in response to climate change.
With temperatures rising globally, our cities become a window into nature's future transformations. Every October in Mexico's Sierra Nevada, monarch butterflies gather in their millions. They traverse the whole of the North American continent to spend winter down south. But our growing cities make their journey ever more perilous.
Monarch butterflies are these really great insects. Unfortunately, right now, they're in decline. In the United States, they've experienced 80% population declines. There we go.
In this industrial area of Toronto, monarch butterflies take a rest stop before flying on. They also take the opportunity to mate and reproduce.
Ooh, it's a really big caterpillar. This is the baby monarch. This one's probably a day or two away from going into its chrysalis and then becoming a monarch butterfly.
While other species have the ability to switch to other food sources, monarch butterflies remain dependent on a single plant.
I got one. Let's check it out. You found a monarch.
The butterflies lay their eggs on the milkweed. Their caterpillars feed exclusively on this plant, and in many cities, the land on which milkweed can grow is disappearing.
Unfortunately, a lot of these cities are providing these barriers that just don't have the resources that they need. And so it would basically be, if you're driving along the road and you don't have any fuel stations and you run out of gas, you're stuck. That's what's happening with these butterflies.
Not all species can adapt. As our cities continue to expand, accommodating wildlife might be crucial. How we shape our cities in the future may prove decisive for the course of life on Earth.
Biodiversity helps us with the food that we eat. It helps us with the air that we breathe. So if we continue along the path that we have, many different populations, including human populations, will start to crash.
Urban evolution can help us design green cities in a Darwinian way.
As humans become more urban, we have the potential to allow some species to live in the city and adapt to our cities, but then put less pressure on the other habitats, which would allow the species that can't survive in the city to continue to thrive.
We're going to see more and more the realization that we are part of nature, and that is actually probably going to help us survive.